As we look forward to the next decade, there are many exciting global contributors which are dedicated to fulfilling the promise of future medicine in a multidisciplinary, collaborative environment like the Broad Institute. In his new role as director, Todd Golub shares his perspectives on the promise and challenges of the future of medicine through a collaborative discussion with Kevin Davis, executive editor of the CRISPR Journal and Gen Biotechnology.
Hello, everyone. Thank you for joining us. I'm Kevin Davis, executive editor of the CRISPR Journal and a new marquee journal in 2022 called Gen Biotechnology, and also the author of the book Editing Humanity, the CRISPR Revolution and the New Era of Genome Editing. I'm honored to be invited to host this closing keynote conversation in which I'm joined by a very special guest, the renowned cancer geneticist and director of the Broad Institute, Todd Golub. Todd, great to see you. Thank you for joining us.
Great to see you, Kevin. Great to be here today.
So let me start by asking you, how are you settling into the new role? What are your priorities? Is there a change in direction at the Broad under your leadership?
Well, anytime there's a change in leadership of any organization, it's really a great opportunity to step back and think about where should we be heading, what's exciting, what does the future look like? And sometimes you don't do that, take every opportunity to do that. And so we're really being very deliberate about that. And from my perspective here, I'm really focusing on my own background as a physician scientist, where, the Broad Institute was from the very beginning intended to be about medicine and not about the genome, for example. It was about understanding the basis of disease and how we could have impact for patients. But I feel like there's a real opportunity now to double down on our commitment to not just write great papers. I think we'll do plenty of that, but to really think about how can we use the power of human biology, human genetics, emerging technologies to really directly benefit patients. So that's going to mean focusing even more deeply on diseases and understanding the mechanism of disease, the root cause of disease, and doubling down on our partnerships with our partner hospitals to work together to take on really important challenges.
Wonderful. We hear a lot about precision medicine. You're one of the leaders in this field. And we've obviously you've obviously helped make great progress in this area over the last decade or so. An almost impossible question, but what do you think will be the big breakthroughs and some of your big priorities that will bring transformative change to precision medicine over the next, say, 5 or 10 years?
Well, we have certain diseases. I'll take major psychiatric illness as an example, where for many decades, we've been stuck with the fact that we've had no insight into the biological basis of these diseases, schizophrenia, bipolar disorder. And so it's not surprising that it's been really hard to make any progress on therapeutics or diagnostics for these diseases. But I think what the next decade is going to reveal, largely being driven by human genetics, is the molecular basis of these and other common diseases that we've never had before. And that's going to be really exciting. I think we'll have an explosion of new mechanistic insights into common diseases that will point for the first time to new therapeutic directions. It's not going to make it immediately easy to turn those biological insights into therapeutics, of course. But in the absence of those mechanistic insights, it's hard to know even where to start. And so I think that's really exciting. If you couple that is the new biological insights into common diseases with the power of gene therapy and genome editing and the ability to deliver these kinds of reagents to patients, increasingly being able to target them to a cell type of interest, that's going to be really powerful because that will make for a whole new generation of therapeutic approaches that we've just never had at our disposal.
Where will these mechanistic insights come from? Is there a certain underlying technology or platform that you're particularly excited about?
Well, I think it's be good to step back a bit first and think about what have the bottlenecks been until now. Until now, it hasn't been possible to do systematic genetic analysis to the point that you could, for example, discover genetic variants that are associated with disease risk. We now have that, or are on a path to having it at the scale necessary to find those disease associations. But there's a whole new bottleneck that we're facing, which is how do you turn those genetic variants into biological understanding? Even if they're coding variants, that is in protein coding genes, they don't come with instructions that tell you what that actually means in terms of disease pathogenesis.
Right.
So I think what we need to do as a field is to invent a whole new set of approaches and tools that will allow scientists to translate genetic variation into mechanistic understanding. Will there be a killer app, a single technology that will make that possible? I don't think so. Certainly genome editing tools deployed at scale, functional genomic methods for base editing and CRISPR knockout are going to be hugely powerful. Machine learning tools also will be particularly useful here. So I think it's going to be a constellation of increasingly high throughput mechanistic functional genomics, functional biology approaches.
Yeah, you've got a few fairly talented genome engineers at the Broad Institute, but I'm glad you mentioned machine learning and AI. How are you betting big on AI being a key component to this sort of next wave of mechanistic understanding?
Yes, we're betting big on the intersection of machine learning and biology being very fertile and hugely exciting. I mean, to make real progress there, you need to be able to generate data at scale. And now we're able to do that in increasingly interesting and sophisticated ways. And you need the ability to create advanced algorithms and to be able to compute in a facile way, typically on the cloud. That's all coming together in a way that we've never had before. At the same time, I do think that there's a lot of magical thinking around this intersection of machine learning and biology. I don't think it's the case that, if you just take a large, database and some smart computer scientists and roll them all together, that some miracle will occur and deep insights will come out. I don't think they will come out automatically. But I do think that if computational scientists and wet lab biologists can together define the problems of our age that are ripe for bringing machine learning to bear, that's going to be really amazing. And so in a new center that we just launched this year called the Eric and Wendy Schmidt Center, is going to do exactly that, to bring computer scientists and biologists together around the world to focus on what are the right questions to answer together across these disciplines. And in many cases, I think it's going to be the computational scientists telling the biologists, here's the data set you should be generating. As opposed to what typically happens is that, the biologists have a data set lying around and they come and find a computational and say, help me, make sense of it. And it's too late.
Right. The Broad, as everyone knows, sits in Kendall Square, arguably the mecca of biotechnology in the world, surrounded by pharma companies and biotech giants and startups. Your faculty are spinning off companies. You've launched many companies over the course of your career. But do you think there is enough collaboration between academia and the biotech industry? It seems like there should be, but I wonder if that is the case.
Yes and no. I've been thinking a lot about this recently. I mean, of course, there's been a long history of collaboration between the academic scientists and industry. But typically that's taken the form of an academic lab takes a project, they take it kind of as far as they can, and then there's a great handoff to a company, which then takes it over, a lot is lost in that handoff. And I think that's not the ideal way. There's some projects where that kind of licensing, more transactional kind of collaboration does make sense. But I think increasingly for the future, there's going to be an opportunity and a need, if we want to really maximize progress, for companies and academia to jointly take on projects, to jointly identify goals that are shared by both organizations, and to say, hey, we could solve this problem more effectively together than individually. Let's create a joint team to do that. And so I'm excited about those kinds of partnerships. We have a number of them at the Broad, but I'd like to see more of them for the future, where there are joint teams where you, in many cases, can't tell the difference between an academic scientist and a company scientist because they're rowing together toward a common goal. Of course, there are important things to look out for, pitfalls to look out for there: conflict of interest, financial conflict of interest, ensuring that trainees, students, and postdocs and so on are allowed the freedom that they need to publish and explore, directions as they see fit. That being said, I see great potential for more joint undertakings.
Yeah, the Broad, as we've touched on, has done just a stupendous job over the last 10, 15 years of developing genome sequencing technologies and analysis methods, genome editing, analysis of common diseases. It's easy to sort of get swept along by all that genomics can do to understand and lead to better treatments for common and rare diseases. Is there a limit to what genomics can tell us?
For sure. And I think what we don't know yet is what is that limit. So I think certainly, as I said before, I think we're now at the point where we know how to go from genomic characterization or genome sequencing. I mean, we're looking at probably $100 whole genome sequence in the not too distant future. The only problem with that is that we don't know what it means. And so I think what the genome can't do yet is explain itself. And that needs to become our job to figure out, for example, what is the clinical utility of whole genome sequence? That's unknown. We don't know, I don't believe, whether that's going to be massive or whether it's going to be quite limited.
Yeah.
And the only way to figure that out is going to be to generate data at sufficient scale, connected with patient clinical information to be able to make those kinds of associations.
We worked almost 2 decades ago on a conference in Spain, which I have fond memories of, called the Molecular Taxonomy of Cancer. And this is not to flatter you, but clearly our progress in understanding the genetics of cancer with cancer panels and sort of making cancer a more personalized diagnosis, it has been a great success story. I wonder, can you sort of try to encapsulate how far we've come and where you're still frustrated at the where perhaps this sort of approach is not helping us in certain types of cancer?
Yeah, well, at the time of that conference, 20 years ago or so, we were at the point where, in very specialized circumstances, specialized cancer centers connected to specialized genomics, research labs, you could do some genomic characterization of a tumor and reveal stuff about it. I think we didn't really anticipate how ubiquitous that kind of approach would become, such that I think it's fair to say today that some degree of genomic characterization of tumors is or should be totally routine for all cancer patients. That being said, it's not the case that most patients diagnosed with cancer benefit from this cancer precision medicine kind of rubric. I don't know what the percentage is, maybe 20% benefit from it. So I think that means that there's a lot we don't understand about what the cancer genome is telling us with respect to predicting therapeutic response. And there are a whole lot of medicines that need to be invented to combat the aberrations that we can see are there. And until we have both those things, I think the cancer precision medicine vision will not be fully realized. But I don't think it's the case that, oh, because it doesn't work for everybody today in 2021 or 2022, it means that the concept was wrong and we should move on to some other concept. I think the concept is right and it's going to be the answer, but there's still a lot of work to be done.
I'm glad you touched on the subject of therapeutics, Todd. In 2021, there were a number of headlines, big headlines, some big papers about progress in finally developing drugs for so-called undruggable cancer targets. Are you excited by some of the progress that's being reported in finally getting to grips with some of these most notorious oncoproteins?
I think it's hugely exciting. I mean, I've never liked the term undruggable.
Yeah.
I think a better term is unprecedented.
Okay.
And I think, the recent progress on a number of previously intractable targets is really exciting. It's going to be critical that we focus drug discovery capabilities on what the human biology is telling us are the right targets, not what the target classes of convenience are. And if the targets that are being revealed by human biology and human genetics turn out to be unprecedented and difficult to target, we need to be committed to going after them anyway, because I'm not optimistic that by avoiding them entirely and simply focusing on the well-trodden path of well-precedented targets, I don't think that's going to be good enough. So I think hopefully this will get people, the recent success in oncology will get people excited and open the lens a bit more.
Yeah, well, it's always notoriously kind of silly to make big sweeping predictions, but I'm thinking of in sort of the lens of 20 years ago, we were sort of celebrating the completion of the Human Genome Project, and look how far we've come in the last 2 decades. What are some of your big hopes, personal and for the Broad and for the scientific community over the next 10, 20 years?
Well, I think the big hopes that I don't think are crazy thinking are that we will have over the decade ahead a first draft understanding of the molecular basis of all common diseases, and that will give way to a new generation of therapeutic approaches that in large measure, enabled by genome editing technologies and other types of gene therapy approaches, will make for therapeutic discovery that looks nothing like what has come before, which, drug discovery today is a very artisanal, one at a time kind of approach, which is shocking that it works at all. It's so difficult.
Yes.
But one can imagine that the ability to deliver genome editing approaches, other kinds of gene therapy approaches, nucleic acid-based therapeutics in sophisticated ways that detect cell states and become activated or delivered only to cells of a particular state, a disease state, for example, that's going to make for a new kind of programmable therapeutics, modular therapeutics that is generalizable in a way we've never seen before. COVID vaccines are maybe the first obvious example of that kind of thinking that I think are really exciting.
Yeah. Well, Todd Golub, Director of the Broad Institute, thank you for your time today. Congratulations on becoming Director of the Broad, and best of luck to you and the Broadies going forward. Thank you for your time.
Great to be with you.